It's 7:01 in Bodenham, and we'll get going. My name is Ed Friedman. I chair Friends of Merrymeeting Bay. Thank you all for joining us this evening. And we are recording tonight. So if you don't want to be recorded, you should leave. Martin, I don't know if you can let that person in while I'm chatting, those people in. I want to thank Martin McGraw, who's here on the call. For organizing our Zoom and being our primary web organizer as well. And Chris Aeppli, who is our presenter tonight. So if you've got any questions, please use the chat feature and I will try and moderate them at the end of the presentation. And we'll go from there. Hopefully we can answer any questions folks have. This is our, excuse me, our 29th year of doing Winter Speaker Series, and this is the fourth presentation this season. Our next one in February will be about noise, community noise, which is getting to be a bigger and bigger problem, along with PFAS and all kinds of other things. So that will be Jamie Banks, who's the founder and president of Quiet Communities. The name of the program is Quiet Equals Healthier. So it should be good. So we're going to go through a few slides here, tell you about who we are, what we do, for those of you that don't know, and then I'll introduce our speaker, Chris. So we are hopefully looking at the duck slide. Is that what we're seeing? Martin? Chris? Somebody?
Yes, that's what we're saying.
Okay, thank you. All right. So we are a unique organization in that we take a holistic look at the environment, looking at sort of these four main areas: research, advocacy, land conservation, and education. And slide on each. A little bit older slide here because it doesn't have PFAS on it. But we've been doing a lot with PFAS, so that many of you know, in conjunction with the air station, historical contamination, legacy contaminants at the air station, former air station, and just in general. But we let our research inform our advocacy. Typically, a lot of this stuff is fairly cutting edge. First ones in Maine to use cage mussels for biomonitoring, unique circulation study, a unique vegetation and land use study, not featured on here. First of its kind probably in the country, to kind of take historical aerial photography, scan it into a GIS format, made it up with current aerial photography, and then look at, in our case, vegetation changes in the bay and around the bay, and land use changes around the bay. As I say, our research often informs our advocacy, a lot of which is focused on migratory fish passage. And you can see there are a lot of turbine mortality issues, whether you're an eel or you're an alewife or salmon. A lot of work with toxics, fish consumption advisories, and obviously, working on the PFAS issue right now, and working really hard to upgrade the next section of the Androscoggin River, moving upriver. Took us many years, but about 3 years ago, we finally got an upgrade from Warumbo Dam and Lisbon Falls down to the bay. And we're trying to move that now up from Lisbon Falls to Gulf Island Pond. And the reason we upgrade is because there's no backsliding allowed of water quality, without a major hoo-ha anyway. And but the way that's measured is by classification. And so when the actual water quality, which is what's supposed to be measured, is of a higher quality than the current classification, which is the case now, getting Class B water quality, but it's a Class C classification, what that amounts to is room to pollute. You can still pollute and meet the current classification, which is too low. So hopefully we'll probably send down an alert about that at some point here. but the legislature is the only body to change that. And hopefully we will see an amendment to a bill they will shortly be getting on that issue. So anyway, education, historically, lots of work in the classrooms, but also out in the field. And we're back to post-COVID doing a lot of the stuff and getting kids out doing hands-on stuff outside twice a year, predominantly in the spring and fall at Bay Day, and getting kids dirty, getting them away from their devices, and using their tactile senses. So it's wonderful. And we protected well over 1,300 acres of wetland, or not wetland, of valuable wildlife habitat over the years. Our focus is on wildlife habitat as opposed to recreational trails. Working on a couple of important pieces now, and yeah. If you enjoy the presentation, or you know people that couldn't make it, we do record these, as I said, and usually within a few days, Martin will have the recording up on the website. And you find it going to the home page and scrolling down the right side to the education section, and you'll see an appropriate link here for speaker series videos. I think we've got about 12 or 13 years worth of videos on here of the 29 that we have actually had happen. So, and here's the rest of the series. Again, I mentioned that the next one here around community noise, slime main. You probably don't know what that is, but that is about gastropods, snails and slugs and stuff, for which there are actually quite a few invasive ones in the state. And yeah, so these all will be great. And here we are. So Chris, our speaker tonight, is an environmental chemist. He's a senior research scientist at the Bigelow Lab for Ocean Sciences in East Boothbay. And he leads a research program in marine pollution and a testing center for PFAS chemicals, commonly known as forever chemicals. I also like to add the everywhere chemicals. See that more and more. Chris studies the fate of organic chemicals in aquatic environments with a focus on PFAS and on residues from marine oil spills. His current work includes the tracing of sources and pathways of PFAS chemicals in coastal waters, such as Casco and Merrymeeting bays. And his research investigates the physical and chemical processes that control how these contaminants move and how they change and how they affect marine life. And with that, I'm going to thank Chris again for being here. I'm going to stop sharing my screen here. He can say hello and launch into his presentation.
Thank you so much, Ed. And thanks for having me here. Thank you, everyone, for taking your time on a Wednesday evening to hear about PFAS in Merrymeeting Bay and Casco Bay. I'll set up my slides. And
you are loud and clear, and now we see your slide full screen, and so I'm going to mute myself for the moment. All right.
Right. Excellent. So thanks a lot for this nice introduction. So here is what I love to do this next hour, or what remains of this hour, is tell you a little bit about the background of PFAS in the environment, specifically in aquatic environments, and then focus on the aftermath of the Brunswick spill, and on a move then to a broader, just like what sources of PFAS do we have that go into the different water bodies, and try to wrap up maybe 10 minutes before the top of the hour or a little bit more to have some questions and answers. As Ed said, you can start just putting questions in the chat. He will moderate that, or just save them for the end. So before I start, I want to thank a lot of people who were involved in all different aspects of that work. Ed, for hosting me here, and more importantly for working together to get all these like great samples that I'm going to talk about later in the presentation. This is also a very collaborative effort with Friends of Casco Bay. Ivy, Heather, Mike were like very instrumental to get a lot of more of the Casco Bay samples. And my colleagues at Bigelow Lab, especially Hannah, did most of the ground legwork and analytical work to get this data together, also with the help of Caitlin. And a lot of this work I present is funded by EPA, a project that just ended at the end of last year. So a little bit about myself. I'm an environmental chemist, and when you first hear chemistry, maybe this is the first impression you have, is a very complicated periodic table of the elements. However, I decided to make my life a little bit easier and go into organic chemistry, because if you're an organic chemist, that has nothing to do with healthy food. All it means, you only care about very few elements on the periodic table of the elements. So carbon, hydrogen, oxygen, nitrogen, and these are basically the building blocks that produce life. But they're also the building block that lead to a lot of chemicals that humans started producing over the time. For example, hydrocarbons that then leads to environmental contamination and lead to a problem. Later on, people discovered that if we add a couple more elements, specifically chloride or bromide, fluoride, and combine this with like the carbon backbone, we can produce very resilient and chemicals that have like specific functions that we need or that we want. For example, PCBs, DDT, you're probably all well aware of how they ended up in the environment. And if we go a little bit lower in the periodic table, after people figured out that the chlorine was maybe not the best, we started using bromine and used something like, for example, brominated flame retardants that then also started accumulating in the environment. And the newest thing now is move one up and go to fluorine and have the fluorinated chemicals. And if you now go into, for example, a sediment core, so this will be like a picture of water, an underlying sediment, and you take a whole core and analyze all these chemicals in this core, there is some nice data from some publication that show the deeper you go, the more of the PAHs you find, this like oil-related compounds. And if you go down to the chlorinated, you find a peak that's a little bit higher up, meaning this was just the chronology of how people use these compounds. Later came the brominated compounds, and now on top of the sediment, you often find this peak of the fluorinated. So I think it's fair to say we're now kind of like in the age of the fluorinated compounds. And it begs the question to ask whether we learned anything over the time except just moving to the next element, but they often have a similar property as these chemicals accumulate in the environment and lead to adverse effects. So why do we see so many PFAS, or these per and polyfluorinated alkyl substances in the environment? And one reason is because they have just very good properties that we liked for a lot of applications. So one would be they're film forming and they're really hard to degrade or to oxidize, so they're great firefighting foam agents. They're also very good at keeping dirt away, so they were used to coat carpets or couches. They're also very good at keeping water away, so waterproofing outdoor chemicals, waterproofing outdoor gear. They also have excellent anti-stick properties, so think about non-stick pans or coated popcorn bags. So this is how PFAS ended up in a lot of different products. I wrote here product that contained PFAS. There is a big push obviously now to get rid of all this PFAS, but sometimes it's not that easy to really identify where they are. But I think we're on the right track. A little history of the PFAS, and I want to just point out like two important timelines. So in the 50s, this is really where the Teflon pans led to a big production of PFOA. So PFOA is not directly, it's not the same as Teflon, but this PFOA was used to produce Teflon. So this is where this came in. And a little bit later, the second compound that you often hear is PFOS. And that's more of a compound that was used for coating of, let's say, clothing, or for foams, or just like surface treatments. So these two compounds have been the original PFAS that were brought into environment in large quantities. Later on, when people realized there is a problem with this, chemical engineers moved on to the next compounds, as we see in a couple slides, that we now can also find in environment. So once it was found that these compounds are a problem, the production, this led to regulation and also to a decrease in production, especially in the Western world. So this plot shows you how the production volume of PFAS changed. In the blue, it's the Japan, Western Europe, and US. And you see somewhere in the 2000s, there's a sharp decline. However, some other estimations showed that it's not really just a stopping of production, but it might more be just a moving to different places. This graph has not been updated for a while, and I suspect and I hope that overall production of PFAS goes down globally in more recent years. So I mentioned these two PFAS compounds that gained the initial attention. And this is basically the top of the pyramid, PFOS and PFOA. Because I'm a chemist, I have to like draw some structures. I drew them here to the side. It's basically a long carbon chain with like a hydrophilic head group. This is pretty much like fatty acid or like soap. So we have one part that loves water and another part that hates water. And this is basically how they work, and that's what all these PFAS share. So when you go down this pyramid, the names might be a little bit confusing, and you might lose your overview of all the different compounds there are, but they all share this hydrophobic, hydrophilic tail. And often the difference, especially for the top part, is just how big this carbon chain is. If you go then more down the pyramid, chemical engineers started more maybe adding another group somewhere in the middle, or try to make it more biodegradable, or put some hydrogens in it. But the pretty much the hydrophobic, hydrophilic nature is always remains the same. So why do we find this PFAS now in the environment? And one reason is exactly this hydrophobic, hydrophilic nature they have, combined with that they have compared to more traditional pollutants, they're pretty water soluble. So they love traveling through the pathway where water goes through. So if you have industry, often the waste infrastructure is then how these compounds end up in environments. So for example, you have a discharge of industry going directly in rivers, or they produce consumer products that go to landfills. Then the landfill leachate will go through the waste infrastructure to a wastewater treatment plant, and they're not really retained in this and end up in the environment. And just like term that's used here, AFFF, is the aqueous film-forming foams. That's also a big source. When they're being used, they often end up in the water directly. Especially in Maine, where we don't have big industry that produced PFAS, a more common pathway is the spreading of sludge, which is also called biosolids, basically the wastewater treatment solids, and it was used for a long time for fertilizing agricultural fields, but PFAS were highly enriched in this sludge. So this is an important pathway how then PFAS got into agricultural soils, but also being washed out and make its way through rivers and into other aquatic environments. How do humans now get exposed to PFAS? And there is various estimation, and it's all very context specific, where you live, what you eat, what you drink. But a general rule of thumb is that maybe 10 to 40% of PFAS you get comes from drinking water. So because PFAS goes into the water, it can be found sometimes in drinking water. And the remaining 40 to 80% is most likely coming from food. This can be either impacted farms where you get food from, or it could be organisms living in water that accumulate PFAS through that. There is also a probably smaller part where you get direct exposure of PFAS from all the other products where PFAS is in there. However, this estimation, it's still evolving, but generally people agree it's that the thermal absorption and taking it in you is not that big as the other big sources, drinking water and food. And here a slide where we can show a little bit of good news, how the attention of PFAS and the regulation that follows actually led to an improvement. So this is PFAS in the US population serum, like blood serum, and there used to be like a regular measurement of a cohort of people, and you can see that over the years where this has been measured, there is an 80% decrease of PFAS concentration from 1999 to 2020. So this is the good news. The bad news is that detected in more than 99% of the US population. So probably all of us listening to this presentation have some trace levels at least of PFAS in their blood, just because we either are exposed through the water, through food, or through some other sources. I'm not a health expert in any way, but there is some great reports that summarize what are now the health effects of PFAS. So here's a link from the National Academies that lays out mostly like 4 to 5 main health risks that can come if your blood serum level is above something like 20 nanograms per milliliter of blood. So because PFAS looks so similar to a fatty acid, but isn't exactly like a fatty acid, it can mess up with the whole lipid metabolism of a body, so cholesterol and lipid metabolism. There has been some studies showing some decreased immune response to viruses, some risk of certain kinds of cancer, and there has been found a correlation between decreased birth weight and significant load of PFAS in the maternal blood. Let's now come to the two main exposure routes, so drinking water and food. First, drinking water. The EPA is studying and measuring the PFAS in drinking water extensively. The most recent round showed that about 10-15% of large and small drinking water facilities have levels that are above the current maximum contamination level of 4 nanograms per liter. So it's not a huge number, but it's a significant number of drinking water treatments. And this 4 nanogram per liter is a relatively new guideline that Maine just adopted per of this year. There is a transition phase until the drinking water facilities need to fulfill that. But just to visualize what this 4 nanograms per liter or part per trillion means, if you think about the Olympic-sized swimming pool, this is about 660,000 gallons of water. So if you take about 10 of the swimming pools and put the equivalent weight of 1 pinch of salt in it, then you get about 4 nanograms per liter of salt in this swimming pool. So it's very, very, very small concentrations that we're talking about here. But on the other hand, we're talking about this low concentration because at very low concentrations, effects have been observed. To end up my introduction, I just want to also talk a little bit about one side of PFAS removal. So they're called forever chemicals, but there are solutions here to remove PFAS. This often is engineered solutions, and it's often easier for if the PFAS is in water than when PFAS is in soil or sediment or somewhere. So on the very right-hand side is what people just used to do traditionally is just putting it in a landfill. The problem that we saw is that the landfills are not, they don't retain the PFAS very well, and PFAS can make it through the leachate into the environment. So on the left-hand side, there is like one main technique to remove PFAS is just adsorption. Basically, you can have a Brita filter with a carbon filter in it, and this charcoal binds PFAS. Another way is destructing it by just burning it, incinerating it. So, for example, is if you want to destroy firefighting foam, you bring it to an incinerator, and at high temperature, PFAS can be destroyed. And there is also some new technology that's being researched and developed that makes the sorption more efficient or makes the destruction possible at lower temperature. So there is definitely like research around, and PFAS is not just in the environment forever, so there is ways to destruct it. And what can we do to prevent PFAS and uptake of PFAS in us? And I think just getting information, I feel, for example, the Maine DEP has great resource there. I think as consumer, I try to buy PFAS-free products, and it's not always easy to find all that information, but it's well worth effort. And if we worried about exposure, whether we have a potential exposure, it's good to think that the main 2 pathways are drinking water or food. If you suspect, if you think that you might have PFAS in your drinking water because you live close to a former biosolid application site, it's always good to test the water. There is some information from DEP how to do that. And I think in terms of food, as I understand it, the Maine CDC is mostly concerned about recreational angler that fish in areas where there is a contamination, a known contamination. And the Maine CDC has a list of which water bodies are affected by PFAS, and they have some advisories for fishing freshwater fish. Great. So to end this first part, so there's many different compounds. Not all of them are equally bad. Detected and can have health effect at very low doses. And I guess regulation works. It just takes time, but so far we see that the regulation that came up slowly starts showing some effects, but it takes a long time. All right, I want to now talk about some of the research projects and about PFAS in our waters. So Bigelow is a ocean science research institute, and what we realized is there has been a lot of PFAS research done in terrestrial environments. This is how PFAS got first attention when we look at the farms and the impact at dairy farms or water sources, etc. But because PFAS travels so nicely through water, eventually all PFAS ends up in the ocean. And there has been not too much research of PFAS in ocean and estuarine and coastal environments. We did a small survey some years ago, just like from Machias to Portland, took some water samples and measured PFAS. And we could basically detect PFAS in each and every sample at low concentration, but still it's detectable everywhere. And unlike some more traditional pollutants where you might expect a higher concentration in more industrialized areas, more maybe towards Portland, it doesn't really matter whether you're at Acadia National Park or Machias or Portland. You find it pretty much at similar concentration everywhere. So there is, as I said, a relatively limited data on PFAS, and also which sources of PFAS are how important to go into the estuaries is a little bit more understudied. So to know exactly if you want to reduce PFAS, which knob and which faucet should you turn down to reduce this, there was some limited data on this. And we chose to look more closely at the Casco Bay estuary because it's a very interesting system that receives a lot of different potential sources. So first of all, we have the Kennebec River here, numbered 4, which is a huge integrator of water throughout Maine that from the Androscoggin-Kennebec system drains a big part of Maine and brings a lot of water in it. We also have this area 3 where we have the Superfund site of the former air station for former Naval Air Station. We have another area around the more Freeport, Yarmouth area where there have been a lot of former sludge spreading sites. In red triangles, I plotted all the former licensed sites where sludge spreading was licensed. And finally, as number 1, we have more urban, industrialized area with a lot of stormwater and industrial effluent outflows there. So we could hypothesize that looking at this system leads to different sources from different locations. We work together as Friends of Casco Bay, Friends of Merrymeeting Bay, and came up with a station list of about 90 different stations that we wanted to sample, and we sampled them bay-wide 3 times in September, October 24, and last year in June. In addition, then we had on the AFFF spill side some additional sampling that we did. So I want to just show you a broad overview of how it looks generally and then jump more into some more details. What I plotted here is just the sum of 8 PFAS in surface water. So this is all water samples. And the bigger the circles, the bigger the concentration. And the number next to the circle shows you the sum of the 8 most detected PFAS. And what you can see is the Androscoggin River up here, Kennebec River down there. We also sampled more higher up north, I'm sure a little bit later. You see the Portland area, you see some rivers, the beside Androscoggin and Kennebec, the Royal and Presumpscot. You see Harpswell Cove that has a little bit higher circles, and the Portland area. Generally, we found the AFFF impact in the Harpswell Sound, Harpswell Cove area. The rivers are integrators of terrestrial PFAS sources, and they obviously, not surprisingly, showed increased PFAS levels coming in. And as I mentioned before, the Portland area were some sources. Some of these bigger circles are some small streams that go into bigger water bodies, but it wasn't especially high, so that was a little bit surprising. When we now look more closely at the Harpswell site, it just so happened that when we were planning this project, this was before the spill. And when we were basically ready to go sampling, this AFFF spill happened that led to the release of about 1,400 gallons of AFFF mixed with 50,000 gallons of water. And it's the largest AFFF spill in Maine, the second largest in the US in the past 30 years. What then happened at this hangar 4 where the AFFF was spilled is that most of the foam ended up in the stormwater system. And the stormwater system related through a series of retention ponds, this pond AB, into this picnic pond. And this picnic pond flows through Merbrook all the way into Harpswell Cove. So it's a pretty direct way, a couple miles, that goes into Harpswell Cove. There also has been some parts that went through an oil water separator that went then northwards towards the Androscoggin via the sewage treatment facility. But this was, compared to the rest, a relatively smaller part. So we were now interested to look at the effect in Harpswell Cove. And what we did is we had a transect along the Harpswell Cove and went there during a year to regularly sample water samples. And I'm going to show you just a brief overview how this generally looks. We have here the one site, and for this site I'll show you how the PFAS concentration changed over time. This is here weeks post-spill, so we extended this all the way to 1 year past the spill. We also happen to have some samples from before the spill. We weren't sampling that systematically before, so there's a little bit more error bar on the before spill, but this is like a good reference value to see like what was the level before, because obviously even before the spill this was a Superfund site with known sources of PFAS. So what you can see from this first site, the concentration increased from around 150 nanograms per liter, or PPT, to almost 1,000 within 5-6 weeks. That means this is how long it took for the PFAS to travel through the stormwater retention ponds into the mouth of the Harpswell Cove. We now also have the site 2, 3, 4, and 5. If you look at site 2, which is still in Harpswell Cove, the scale here changes, so it goes up to a little bit more than 250 nanograms per liter. But the time scale of when we see this pulse of PFAS coming through is approximately the same. And even if you go further down, site 3 or 4, this is after the bridge at Mountain Road, or then site 5, this is a buoy from Bowdoin College that's out there. You always see a pulse of PFAS coming and then reaching pre-spill levels. So the lowest 3 graphs have, again, a different scale. Here the max was around 40 nanograms per liter for the sum of the 8 PFAS. And at site 5, you see a small effect, but it's still remarkable that we still see this pulse of PFAS leaving. And if you know the Harpswell Cove, Harpswell Sound area, it's very tidally influenced, so it's flushed out every day through this very narrow pathways. There is a little secondary pathway around site 3 where water can escape here, but most of it really goes from site 1 through site 5 back and forth and flushes it out. So in this case here, it really appears to that the dilution that is driven by the tide is really the kind of like solution to reduce the PFAS concentration. We also are currently doing some more fancy statistical analysis to look at, can we match the fingerprint of each PFAS sample, of each PFAS source, and determine how much of firefighting foam impacted PFAS versus background PFAS is there. So what we then do is basically taking some source profiles, something more terrestrial, something more that's in the ocean background, and something more that's a firefighting foam, and match it to the sample, and then for each sample we can then say how much of this sample comes from terrestrial sources versus ocean background versus AFFF. So this is still work that's a little bit in progress here, but it seems to be promising that when we apply this over this Harpswell Cove over time, we have the September, October, and then June 2025. We see that the red pie chart is decreasing over time, meaning the impact from the AFFF is decreasing, so the signal decreases there, which is good. And we can even take one particular station and then just plot this over time. What's the contribution of AFFF over time? And in most cases, it's a pretty reasonable curve that we can observe here. Okay. The next thing I want to talk about now is Kennebec and Androscoggin. When we started this project, we wanted to get samples from the Kennebec and Androscoggin because Ed has been collecting PFAS samples for years in this system, and because it's such an important contributor to Casco Bay. And when I approached him, he said, like, sure, we can sample here, and the best thing will probably be just use the helicopter, because this is going to be like the most efficient way to do that. So I'll show you a couple pictures. I got the privilege to join him for like 3 sampling campaigns, going through the Androscoggin and Kennebec, and landing at sites where we wanted to get PFAS, which, there is a lot of dams and location that would be difficult to access otherwise, and that was like a really nice way to collect PFAS samples. So what we did was basically we started somewhere in Lewiston, went down to Androscoggin, around Brunswick, and then up through Merrymeeting Bay, back to Augusta. And I'll now show you, again in similar pie charts as you saw before, what the PFAS level look like. So again, what you see is pie charts of these different PFAS compounds. So the big ones is the PFOA in red and the PFOS in green. This is like the major compounds that we often found, but also the other one that often have been observed. And the number right next to it would be the sum of these 8 compounds in nanogram per liter or part per trillion. And when you just look at this one campaign here, you see that concentration are not super high, but detectable. And when you look at the different pie charts, you see that the Kennebec and the Androscoggin have their distinct profile. I'll now flip to 1 month later. This would be now the October. You see some change, but not too much. And then I'll go 8 months, 10 months later. This will be the June again. So overall, the conclusion is that we have a relatively uniform concentration along the individual rivers. So whether you go upstream or downstream, it doesn't matter that much in most of the months. it's a distinct profile for Kennebec versus Androscoggin. And if you compare it over the 3 sampling campaigns we do, there isn't a large temporal variation, so it's remarkable constant. To show you again, I'll flip back and forth a couple times. So we have September, October, June, September, October, June. Obviously, both these systems are tidally influenced, so it's not always perfect because you just have to we try to catch it at the same tide level, but you don't always manage to do that perfectly. So this then begs the question, so if this is so uniform, what are the different sources that lead into this river or then ultimately into the bay? And if we think back of what we originally set out to do is say like, well, there is the different influence from the rivers. There could be runoff, there could be stormwater, industrial effluent, and on top of this we had the Aeppli spill. So the Aeppli spill is definitely one source that that happened in the Casco Bay. We also have all the rivers that integrate PFAS, and what we can do here is we basically can measure the concentration of the PFAS, multiply by the flow, and estimate how much PFAS comes in through the rivers. And then going a little bit more into point sources, often wastewater treatment plants are cited as being like a significant input of PFAS into the environment. And fortunately, there's a lot of data from PFAS of wastewater effluents. So we went into the data and looked at various effluents data and flow data to estimate how much PFAS is inputted into rivers and the bay through wastewater treatment plants. So here is a little bit a closer view on the Kennebec as an example for how we did this. So this is the Kennebec, and these are some of the major wastewater treatment plants: Anson Madison, the Kennebec Sanitary District, Greater Augusta, Gardiner, and then Bath. You can look at what their typical concentration is. I here just looked at PFOS as one of the main compound. You multiply this with the annual flow and you get a rough estimate. This is not exact data by any mean, but it's a rough estimate how much PFOS would be put in the river from this particular treatment plants. And then you can say this Anson Madison is about 80 grams per year, KSTD 500 grams. Then you have in Augusta like in the hundreds, Gardiner order of magnitude lower, and then Bath maybe 70, and the BAW just about 2 grams per year. So this is a way to estimate what's the source contribution to these rivers. What you can do now is compare this to the flow and the mass of PFAS from the overall river. So I'll walk you through this. On the very left hand, I just plotted again the BNAS AFFF spill, and we have a conservative estimation is if everything that got spilled ends up in the bay, this will be about 20 kilograms of PFOS that was put into the environment in this one-time event. When we now look at the rivers, we can estimate how much PFOS is put into the system every year, so in Casco Bay. And you see the Royal is about 3 kilograms, the Presumpscot 4, and the Kennebec is then about 16 kilograms of PFOS coming through the river and into the bay. Now, when we compare this to what we know from the wastewater treatment plants, we know it's a very minimal load that we really can account for so far. And if you look at the wastewater treatment plants going directly into the Casco Bay, it's even lower. So what the conclusion there is that beside the wastewater treatment plants, there must be a lot of unidentified sources which is either some diffuse sources, a lot of agricultural runoff, for example. It could be a lot of old landfills along this river, or diffuse sources like groundwater, exfiltration, or atmospheric deposition that happens there. So I guess this is a project where, again, it ends up with a lot of other questions and asking, so what are really the sources of all this PFAS in the rivers? But I will leave you with here and make sure we have a little bit time for question and just say, like, PFAS are prevalent at low concentration. We can try to track AFFF-related PFAS, and the wastewater treatment plants surprisingly seem to be a minor input into the bay. And with this, I want to thanks for your attention, and just shout out to like another event that's coming up in like 3 months. We're still working through the data, but Friends of Casco Bay is gonna have a coffee with the baykeeper where we go a little bit more into the detail of specifically the Casco Bay data. Thanks a lot, and I'll hand over to Ed to see whether there is any questions.
That was fantastic, Chris. I really appreciated that.